Can you hear me all right?
Yes.
Okay, great. So pleased and honored to be here to not only thank you for all of your support over the years, yesterday, today, and tomorrow, but to remind us all that you all are the heart and soul of what makes JDRF tick. You know, we've been at this for 50 years now. Many of you came in just in recent years, and some of us, including our host this evening and her family, have been involved almost from the beginning of those 50 years. Before I introduce you to our wonderful host, I want to do just a couple of housekeeping things. We are going to record this call so that people who weren't able to join us tonight can catch up and hear about all that you will be privileged to hear. And should you have any questions throughout the presentations, please continue to throw questions in the chat, and we'll make sure that they are addressed later in the program. So as I mentioned, this is our 50th year in business, and we've marked it with our tagline, "The Power of Us." And seeing all your smiling faces, it reminds me yet again how powerful we are as a group. Whether you hail from Salt Lake City or Denver or Boston or Texas, in my case, we're all part of this extremely powerful organization and passionate organization. Speaking of passion, there's no one from Boston more passionate than Lisa Wallach and her husband Neil, who I believe I saw him on here as well. I will let Lisa tell you her story, so I don't want to take away her very passionate story. But she's joining us today wearing the hat of Vice Chair of our International Board, having worked in a number of other roles. So without further ado, let me pass the mic to Lisa so that she can get this, she can tell you all about herself.
Thank you, Marianne. And thank you, Hannah, and thank you, Erica. Wow, what a special evening that you put together. And I'm so jealous that I can't be with you all in person and enjoying such an incredible spread for such a good cause. As Marianne said, my story goes back a long way. It begins back in 1970 when my younger brother Scott was diagnosed with type 1 diabetes at the age of 18 months. And at the time, people really didn't know what type 1 diabetes was. We didn't even call it type 1 diabetes. And there was certainly no organization dedicated specifically to this disease, and no one focused on research to find the cure. And that led my parents to be among the early founders to help create this organization. My mom founded the New Haven chapter, and my dad went on to serve on the research committee for decades and to serve as chair of the international board in the 80s. My story got more complicated in 2001 when our son Harris was also diagnosed with type 1 diabetes at the age of 5. My parents were actually babysitting at the time, and they spotted the symptoms that even though I had grown up with my brother having type 1 and being a second mother to him, I hadn't noticed them myself. And sure enough, they were right. And that led to my leaning in and essentially becoming my mother. I have seen firsthand how much safer and easier it has become to live with type 1 since JDRF was founded 50 years ago. We started with squeezing diapers over urine strips, and then there were very rudimentary blood sugar testing kits available. And now we have continuous glucose monitoring and hybrid closed loop pump systems. And both my brother and my son are benefiting from the Tandem Control IQ system, and we are all sleeping much better at night as a result. I hope that many of you are also taking advantage of this new technology. After Harris's diagnosis, as I mentioned, I followed in my parents' footsteps and really leaned in, taking on all tasks, great and small, I eventually served as chapter president of the New England chapter. I chaired our leadership giving effort and our stewardship committee, and I continue to serve on the gala, outreach, and nominating and governance committees. Nationally, as Marianne said, I'm proud to serve as vice chair of the International Board. And I feel truly grateful to have the privilege of committing so much of my time to help JDRF and our mission. My husband Neil and I give as generously as we can to JDRF, and we're very proud to be leadership giving donors and members of the Beta Society. We also use the walk, the ride, the gala, and any program we can to leverage support from our friends and colleagues. My husband Neil has been running marathons and Ironman triathlons for 2 decades, through which we've raised nearly $1 million in support for JDRF. It's pretty amazing what you can do if you just put your mind to it and are not shy about asking everyone you know to support this cause. These are obviously very challenging times for all of us and certainly for all nonprofits, but I am incredibly proud of JDRF's strategic vision and leadership and the steps that are being taken to ensure that our mission can continue. Research progress has never been more exciting. JDRF works across the pipeline, from funding research done by the best and brightest scientists around the globe, to building consortia of scientists from beta cell biology, immunology, and other fields to collaborate with one another, to working with the FDA and industry partners to ensure that new products and therapies will be approved, and to advocating with insurance companies to make sure that they are covered. A key step in this pipeline is ensuring that the exciting new therapies that are developed in the lab can get the tens of millions, if not billions, in funding that are needed to advance towards clinical trials and on to commercial development. To enable this to happen, JDRF created the T1D Fund, a venture philanthropy vehicle which uses donor dollars to catalyze and accelerate a market for T1D therapies. Neil and I are also proud to be donors to JDRF's T1D Fund. On behalf of the International Board of JDRF and everyone who has been impacted by T1D, I want to thank each of you for your partnership and your investment in JDRF and our shared mission to conquer this disease. Today's discussion will give you the opportunity to hear about some of the exciting progress made possible thanks to JDRF and your support. JDRF-funded researchers and clinicians, Dr. Peter Gottlieb and Dr. Aaron Michels, are joining us this evening to discuss their work to develop personalized therapies against genetic targets for autoimmune diseases, including T1D. Many of you, especially those based in Colorado, will be familiar with Dr. Gottlieb and Dr. Michels, who are truly T1D celebrities, especially in the Rocky Mountain region. In addition to being dedicated endocrinologists at the UC Barbara Davis Center and brilliant scientists pursuing therapies for T1D through their company IM Therapeutics, Doctors Gottlieb and Michels are extraordinary examples of the role JDRF plays in accelerating cures for T1D. JDRF is proud to have provided both of tonight's speakers with numerous research grants over the course of their careers. And when they were ready to launch IM Therapeutics to pursue their approach, JDRF, through the T1D Fund, was thrilled to partner with them to develop their business model, lead their startup financing, and even relinquish their entrepreneur in residence to serve as the company's CEO. Moreover, the work of tonight's presenters is deeply personal. Dr. Michels himself lives with T1D, and his daughter was diagnosed with T1D at the age of 5. And Dr. Gottlieb's mother lived with T1D, and he has 2 children with the disease. Thank you both for all that you are doing for our shared cause, and please take it away.
Thank you very much, Lisa. Aaron, can you get your slides up?
Yeah, I can.
Okay. Well, thanks everyone. We really appreciate the time, and this is going to be pretty interactive. So we have some slides that will talk a little bit of how we came to where we are. And certainly feel free to either ask us questions while we're doing this or as we continue on. And we'll try to sort of let you know where we think we can go with with really trying to make an impact on the disease. So we named the company Immunomolecular Therapeutics, but it's sort of hard to say. So we call it IM Therapeutics, or IMT for short. And this is sort of our tagline, redefining autoimmunity, transforming lives. And really what we're after and what we're going at and what we're going to explain tonight is that we're building personalized medicine for autoimmune disease. And we'll try to explain why we think that's really, really important. Let's go to the next slide. Thanks. So, as Lisa mentioned, we've had funding from JDRF, and I can tell you that my first grant when I entered the field many, many moons ago was a JDRF fellowship. So we've been funded. And I think one of the things that JDRF realized a number of years ago was that you can get really good ideas going, you can really support really good people, and you can get things to a certain stage within academia. But at some point, that idea, that concept, that thing that you want to do, where do we want it to go? We want it to go into people. We want it to treat our kids, our other loved ones. So that's really something that was a gap that existed in trying to move from an academic setting, from a really good idea into something that could form a company and then obviously move forward from there. And we recognized that as well several years ago, work that Aaron's going to explain to you that he was doing with George Eisenbarth at the Barbara Davis Center, that really was building on an idea that probably was 20 years in the making, to be really honest. So these things, they take time. They just don't happen overnight. But we got to the stage where we realized we had done a lot of really good things, and we'll tell you about some of them in the slides coming up, but that we really needed to, if we wanted to go further, we really needed to start a company to be able to take it into human subjects, to take it to clinical trials, and hopefully in a few years to a therapy that might be approved for type 1. So we started the company then. As many of you know, this is a big market and it doesn't have an approved immune therapy. So it's something that in theory, we hope that other pharmaceutical companies and biotech companies will be interested in. As we're going to argue tonight, we think that if you can take type 1 diabetes and realize that it's not the same disease in every individual, that some people go down this pathway to get disease, some people go down that pathway to get disease. And what we want to do is actually go after the pathway that they're using. And by doing that, we think we can get a personalized medicine approach that can actually be more effective. And that's what we're going to explain this evening. This approach, we think, won't just work for type 1. We think it can work for other autoimmune diseases like celiac, and we'll talk about that a little bit. The lead drug that we have so far, we've called IMT-002. It blocks one of the HLA human lymphocyte antigen genes, DQ8. And we've just completed our phase 1A clinical trial that went really well. And despite COVID and all the rest and stuff, got that done and finished that in June. And we've just actually launched our phase 1B. We'll give you a little more details about that. So we're working very strongly on type 1. As you already heard, Aaron and I have a really strong emphasis on that. And I think one of the things we really appreciate about the T1D fund as sort of bridging us over from the JDRF funding we had previously is their focus is on type 1 as well. So we really want to go after type 1 first. And certainly if this paradigm works for this disease, move it to other ones as well. And that's the pipeline that we'll mention in a moment. So far, the way this has worked is that we've secured what's called Series A funding. I'm learning the jargon, so I'm getting better at this. Aaron and I are really good scientists. I can't say that we're the most amazing business people around. Some of you might be far better at that than us. So, but we're learning. We may not be as quick studies on this as some other things. Thankfully, we've raised about $20 million so far. A lot of that money coming from the JDRF T1D fund from Morningside Ventures. And in addition to some venture some angel funding, as well as SBIR and other Colorado grants, our total that we've raised to date is $24 million. And next slide. So this is just a really quick overview, and I don't think we'll spend too much time on this, but really the idea is that we're really trying to go after unmet medical needs. Type 1, as I said, and as we all know, insulin really helps. Pumps are good. CGM and closed loop systems are really clearly doing better for people overall. But if we could really prevent the disease process from moving forward and taking out the insulin producing cells, we could do much better for our, patients, our family members, et cetera. So that's really the goal of our therapy. The individuals who get type 1 a year, it's anywhere from 50 to 60,000. Of those, 60% have this particular gene called HLA-DQ8. About 30 to 35% have another HLA gene, DQ2, and we'll talk about that because between the two drugs that we're developing, we actually would cover most of type 1 diabetes. So we think that this kind of therapy could help someone who's still producing their own beta cell, who's still producing their own insulin from their own beta cells. Certainly, if we can get it into them even before they need a lot of insulin, it would even be more effective. And that's sort of the approach that we're going to take. DQ2 not only affects diabetes, but it also affects celiac disease. And because of that, that's become another focus for the company. It's probably one of the reasons we've gotten the other venture capital people, drug company people that we've been speaking to interested in us as well, because we're actually looking at two diseases. And from a business point of view, as you might imagine, they like that. Since a lot of people have celiac along with type 1, it obviously is something that everyone's aware of. There's, again, no approved drug. It's a very large market, as you can see. And flipped a little bit from type 1, 90% have DQ2, only 10% have DQ8. But again, as you can see with our approach, we can develop one drug to fit one disease, but then actually have an effect on another. And that's sort of one of the advantages. As our company grows and as we hopefully get more funding, we're going to look to expand to other diseases, and that's what's listed below. But we won't go into too many details in that tonight, but if there are questions about it, feel free to ask us later. Next. All right, so just to sort of just put things in perspective for where we're sitting today, and this was not the case when Lisa's brother was diagnosed or when my kids were diagnosed, but certainly we've made a lot of progress in where things are now. So as Aaron and I would both tell anyone, this is a predictable disease that we can measure type 1 diabetes antibodies. We can tell where someone is sort of on that spectrum and can follow them and potentially even intervene earlier before the full elevated blood sugars occur. Unfortunately, there's an increasing incidence. It's not just in the US, it's worldwide. And so there's really an impetus to really push on this. Different pathways lead to disease development, as I've sort of been mentioning, and we're going to show you a little of the ideas for that. And we really do believe that immune intervention can impact the disease. Teplizumab, which works in a very different way from what we're talking about, obviously can delay diabetes onset in the recent TN10 trial that was published in the New England Journal. And really, I think promises an approach that can be helpful. But again, as we'll speak about tonight, we think that our approach can complement that, but also really is very safe, very specific, and very personalized, which I think should be a benefit to individuals down the road. Next. So I'm going to turn it over to Aaron since he's focused on this and try to walk you through the scientific rationale for how we got to where we are and where we hope to go.
All right, thanks, Peter, and thanks everyone for joining tonight. So do feel free to interrupt and ask questions. As you saw, there's a lot of personal involvement here from Lisa, Peter, myself with diabetes. And just to highlight some of those other points, my daughter, we knew she was going to develop diabetes. She had all of the markers at the age of 2. Unfortunately, she was 5 and not a little bit bigger. But these things do happen. And you know, our goal here is to cure diabetes, to get rid of diabetes. Technology is great. I use it too. My daughter uses it. Many patients use it, but I want to get rid of it. So that was a lot of the thinking 10 years ago when we said, you know, we know a lot about type 1 diabetes. Why are we still treating people with insulin? It doesn't do anything for the disease. It's needed. And so, you know, in the 2009, a lot of genetic studies were done. These were taking thousands of people with diabetes, thousands of people without. And they looked at many genes, and this just shows the risk for developing diabetes. So if you had certain HLA genes, you could be 6.5 up to 10 times more likely to develop diabetes than someone else. And the next one was insulin. And this is not like insulin in the metabolic fashion that helps us lower blood sugar, but it actually leads to insulin expression in the thymus and education of specific immune cells. And so it was pretty clear, to me at least, looking at this list, And type 1 diabetes, these are the major genetic risk factors. These probably underlie significant pathology in type 1 diabetes. So all genes make proteins. So what do these genes do? We knew that this DQ8 gene was in 60% of everyone with diabetes, 60% of those at risk. If you had it, you were 10 to 11 times more likely to develop diabetes than not. These genes make proteins on specific immune cells. These cells that have DQ8 take up protein, so think insulin, and they activate T cells. And we know that DQ8 presents insulin peptides and activates CD4 T cells that remain in the islets of individuals with type 1 diabetes. So providing just a good framework for pathogenesis and what's going on in at least a select subset of patients with type 1 diabetes. It shouldn't be too shocking that we wanted to look at DQ8 and say, how can we target this? How can we specifically target it, block its function? And if we can do that in a safe and specific manner, will it do good in type 1 diabetes? So conceptually, this is that DQ8 molecule. Here's the immune cell that gets activated. We wanted to take a drug, put it right in here, so you could not present this piece of insulin and activate the T cell. We thought this would be great if it would work. It was a good idea. And so about 10 years ago, we set about doing that. We started with a supercomputer. We took the structure of DQ8 and said, how can we put drugs in there? How can we put small molecules? We developed benchtop laboratory screening tests to see if some of those compounds would work. We did use animal models. There are very good animal models of spontaneous diabetes. And eventually we decided to go into people and say, will this work? Can we block DQ8 in people with type 1 diabetes and have an effect? And we'll go through some of that. So to start, we used a supercomputer, and this is DQ8. And there are these little pockets, there's 4 of them along here, that really bind small molecule drugs nicely. So think of things like Tylenol, aspirin, and they just fit right in there. And so we took a library of 140,000 compounds, put each one in there in 1,000 different orientations, and said which ones are predicted to occupy and bind. We did that. We then took them to the bench. I'm not going to show you all of that fun work, but we found compounds that worked. We took one of those into this mouse model of diabetes, drug shown here. When we give the drug, we delay diabetes onset early in disease. Later in the disease, we're able to maintain normal blood sugar, actually very similar to that drug anti-CD3, which was just shown to delay diabetes incidence. And when we looked at the pancreas, shown here, brown is insulin staining. There are nice intact islets, and these mice are able to maintain blood sugars and not develop diabetes. This was all good, but this is mice. So we then asked the question, there are about 3,200 drugs already approved, out and available. These are Tylenol, Advil, antibiotics. So we screened all of those and said, can we get lucky? Can we find something that may interact with DQ8? This is how we found methyldopa. It is one of the very first antihypertensive drugs used to lower blood pressure. It's been in clinical use over 50 years. It's on the World Health Organization of necessary medicines. Its main role today is to treat pregnancy-induced hypertension. That again should speak to its safety. So that was good to see. We do not need this at all for the blood pressure lowering effect, but it does bind DQ8 and it blocks these insulin reactive T cells and other DQ8 T cells that are activated quite nicely. Again, a lot more work was done, but Peter and I have this MD behind our name, and we really wanted to know, is this going to work in people? Is this going to hold water? All the mouse studies are nice. All these elegant, fancy experiments you can do on the benchtop are good. So we went back to the JDRF, who funded a lot of our initial work, and said, we're ready to do a clinical trial. The JDRF was nice enough to fund this. And this is actually even before we formed the company. This is still Peter and I working at the Barbara Davis Center in academia. So we had methyldopa, a known drug, not used for diabetes, but we took type 1 diabetes patients at the center that were newly diagnosed that had DQ8. So they're able to make some of their own insulin, and we gave them drug in doses that are used to lower blood pressure. And we kind of dose titrated over several weeks and then washed out. Remarkably, this worked well. We set up assays using the patient's blood to show that we could block DQ8 in their peripheral blood immune cells from presenting antigen. Blue is on drug, red is off drug. So the effect is reversible. When the drug was withdrawn, it came back to normal. And this works for things like insulin and gliadin. It's an antigen in celiac disease. We're able to block that as well. This is very nice, but we wanted to be specific. We have a number of HLA molecules. Most of us probably have about 12 that are on immune cells. Other ones that are quite common, this one called DR4 and DQ2, didn't change during treatment. However, DQ8 did. So that was very nice, showing that we can block the function of DQ8 in peripheral blood. There are also metabolic endpoints that we look at all the time, and many of you familiar with diabetes will know hemoglobin A1C. Our 20 patients after 3 months look pretty good, 6.5%. This didn't come at the expense of more insulin use per kilogram of body weight. And then the gold standard for kind of saying, hey, are you still able to make some of your own insulin, is measuring C-peptide after you receive this mixed meal tolerance challenge and just calculating an area under the curve. And what was really nice to see is that we maintained. Some people did go up, some did go down, but really the goal is to keep people making their own insulin. So with this as a backdrop, this was very good. And we were able to form and start the company. We also did studies looking at the individual T cells within the patients. So there's different types of immune cells. There are those that have DQ8, and then there's the cells that become activated by DQ8. And so we know that insulin and certain parts of insulin activate these T cells. You can see in all of the patients that had a response at baseline, after they received drug, that those inflammatory T cell responses decreased. This was not the case for tetanus. Many of us are vaccinated against tetanus. So again, very good to see that we are blocking what we want to and leaving normal immune function intact when we give a drug to block DQ8. I should say methyl-dopa is a good drug. Patients were able to tolerate it. There was a minimal to no blood pressure lowering, which was good to see. So otherwise, young adult healthy patients with type 1 diabetes didn't lower their blood pressure. But it's not ideal. The half-life is very short. We had to have people take the drug up to 3 times a day. And we really just wanted to do better. We did not need this drug to be metabolized and lower blood pressure. And so we used a nice trick of nature and how the body likes to use amino acids and build. And we switched L-methyl-dopa to D-methyl-dopa. D-methyl-dopa is not metabolized. In studies we did, it was much more effective than aldomet-methyl-dopa in blue, and has a longer on rate for DQ8. And since it's not metabolized, half-life looks a lot better as well. And really this is a lot of what led us to selecting this as a lead drug in our company. Other really nice things about this, this drug and our approach. So the immune systems that we have are there for a reason. Obviously, to fight infection, it becomes abnormal in those of us that have type 1 diabetes, celiac, and other autoimmune diseases. But we really wanted to just block things like insulin and gliadin. We call those kind of self-antigens. But if you got influenza, happens a lot, we'd really like those responses to be intact. And so we set up a system to test this in a laboratory assay. And so with our drug, we have a T cell that sees a piece of influenza presented by DQ8. We are not able to inhibit that. If we use higher affinity compounds that bind a bit stronger, we are able to block that. And if we actually use antibodies that bind to DQ8, you can see an inhibition of that response. And so what this really does is we're giving a drug, we're able to block what we want very specifically, but leave responses to, as I showed earlier, tetanus, toxin, and influenza intact. And I think this is really important. A lot of us have kids with diabetes. You know, if you have a 5-year-old, are you going to just knock out an arm of their immune system, and are you going to feel okay about that? That's a very difficult thing to do. And so I know we've kind of gone through this fast, but this was always a priority in the development and thinking, really over the last 10 years as we were thinking about drugs and what we would want them to look like. I guess, Peter, I'll turn this one back over to you. The current clinical trial we have going on right now through the company using this drug IMT-002 in DQ8 individuals with type 1 diabetes.
So thanks, Aaron. So the money that we received from that Series A that included the funding from the T1D fund allowed us to do all the sort of preclinical work that you need to do in animal models to show the FDA that this is a safe medicine to give to human subjects. And we were able to complete all of that through the course of the last year or so, and actually submitted what's called our investigational new drug application to the FDA in April. And then it was approved 30 days later. They had no questions for us, which was good. And then we actually did what was called a phase 1A study. That's where we gave a single dose of the medicine. We're really looking for safety and tolerability. These were to adults, but again, not people with diabetes and not necessarily even people with DQ8. We just really wanted to see, was the drug safe when we gave it to them, since it hadn't really been given to people in over 50 years or so. And also, we really wanted to look at that pharmacokinetic profile that Aaron had shown you before. What was very good about things was that even with COVID, we were able to get the study done very quickly over the course of a month, treated all 4 of the different groups that we treated, and then got the data back about a month later. First off, no adverse events whatsoever, even at the maximum dose that we used. What we saw from that pharmacokinetic analysis, the PK profile, was that the drug looked really good. It has a longer half-life than the L-methyldopa, the commercial drug that's available, and we think might lead to us being able to just dose the drug once, at the most twice a day, which would be an advantage. So this phase 1B study is designed to go into people with type 1 who have DQ8. So we're screening them for that particular gene. If you don't have the DQ8, then you can't go into the trial. And we're going to dose at these various different levels. We're testing 350 twice a day. That's what the BID means. 1050 once a day, because again, as we just talked about, maybe we can get away with once a day. 700 milligrams twice a day and 1050 twice a day. In these early studies, we really want to do a large range of medicine to be able to sort of get a good feeling for what's going to be the right dosing level that we're going to use in a study that we're going to do for a lot longer that's called a phase 2 study. That will be our first really true look at efficacy. For this study, we're going to treat individuals for 2 weeks and look at a lot of the safety, the pharmacokinetics, but particularly what you saw when Aaron was showing you the work we did with L-methyldopa, that blocking assay. It's going to be very important. We want to see at what level do we get the best blocking with these different medicines. So we're doing this at 3 different sites to try to, again, mitigate for COVID. And we're halfway through that first cohort as we speak. So far, so good on things. We'll have to see how things go with COVID over the next few months. But we're hoping to finish this in around March, be able to run those assays to understand how the medicine has worked in early April and May up to June, and then basically be set up to do a phase 2 study in early 2022. And that's really the next approach that we're going for diabetes, is to try to do that study that would allow us to optimize dose, hopefully go into young kids. And that's something we'll have to discuss with the FDA. As Aaron mentioned, this is very safe, and that's one thing that's a good thing, but it is slightly different from the other medicine, and the FDA does consider it a new compound, which sometimes makes them, asks us to show them some safety before we move the age down. Ultimately, we'd like to be treating as low as 6 to 8 years of age for that phase 2 study. And that would be one that would set us up, hopefully, to really understand how the medicine works. I think there are other things we could talk about for the company, but I don't know if at this point it would make sense to see if there are questions that people have or where to go from there. And as was mentioned, look, we started with JDRF because of the T1D fund. We were really able to open doors and get more funding for the company. And the T1D fund is continuing to be a active partner, as was mentioned, and really will be part of us as we work our way through. In the next funding cycle that we'll do is called a Series B. That will be next summer, and JDRF or T1D fund will be part of that as we really try to keep moving this forward. And since we all have that focus on diabetes, that's something that I think Aaron and I are very thankful for. Want to add anything?
I just wanted to share that there's no questions in the chat yet. I think that folks are starting to ask them, but Lisa, will you share, because for a lot of folks on the phone, this might be their first time hearing about JDRF's T1D fund and this concept of venture philanthropy. Will you share a little bit more about what makes this so unique and some of the ways that JDRF is really changing the space in venture philanthropy?
Absolutely. It's not the first venture philanthropy fund, but I think we're the most successful to date, which is amazing. The idea here, just to be more clear, is to basically be raising private dollars from donors. It's not an investment where one gets a return. It is true philanthropy. Including the dollars that JDRF itself has invested in the fund. And the goal is to use this money very strategically to essentially give a little bit of a boost and an opportunity for new companies like IM Therapeutics to attract the kind of private funding that is really what's needed to enable again, as I said, in many millions of dollars needed to develop products and move them into the commercial space. And Sean Doherty, who is also from Boston, a dear friend who is kind of the volunteer lead of the T1D fund, frequently will say, we would never want to, as an investment, put all of your dollars into something as specific as T1D. But that is exactly what we can do as a venture philanthropy fund. And by giving a little bit here and a little bit there, essentially creating a market for other companies, other investors to want to come into this space. And as was said, T1D is one of the diseases that does not yet have any therapeutics available. Same with celiac. And I think it's an interesting comment that was made that to the investors, the private capital, having the ability to have the drug and therapy impact more than just T1D was incredibly attractive and has encouraged more dollars to come into the space. So any money that is made on any of the investments as these companies continue to be bought and do well comes right back into the T1D fund and is utilized to fund additional investments. And our hope and dream is to be able to have this thing running on its own profits. If I say profits, again, going back into the fund in the near future. But it's incredibly exciting, and it really has transformed the marketplace and filled that gap that's been out there between basic research, exciting research in the lab, and hopefully the market. I hope that answers the question. And happy to take other questions if people want things more specific details.
I'm sure we'll get some more questions coming in. We do have a question for Dr. Gottlieb and Dr. Michels. Would you eventually study kids with celiac, but not type 1 diabetes? Our family has one of each.
Aaron, you want to do that one?
Sure. Yeah. So the quick answer is yes. And so obviously this all started with type 1 diabetes. But with a lot of that, again, generous funding, the company, it really does accelerate progress. And so we are building drugs for DQ2. And DQ2 is in 9 out of 10 people with celiac disease. And our goal there is eventually to use it in type 1 diabetes, but we will start in celiac disease because it's so common, and it really does drive that disease. I mean, it drives those immune cells that target and destroy the small intestine. And so clinical trials, if all stays on track, will again start in probably 2021, the later half of the year, in celiac disease with a drug that specifically targets DQ2. We're just a little bit ahead in type 1 diabetes. But it is true, out of all of our type 1 diabetes patients, 10% have celiac disease. I'm sitting here, I have both as well. Yeah, neither one of them are great to live with, quite honestly. And together, it's even more of a pain. But our initial approach really was to go after DQ8 and DQ2. We can essentially then be able to treat, maybe with more than one medicine, everyone with type 1 diabetes and celiac disease.
Love hearing that, and love the nod from Dr. Gottlieb too. Another question, for Dr. Gottlieb and Dr. Michels, if you can share your thoughts on the stages of type 1 and which stages would be optimal to study moving forward, and if that would be similar to teplizumab.
Sure. Let me start with that, and then I'll let Aaron add a little too. So the part of where we're sitting a little bit is that in general terms, we have to think about the business aspect. And so as companies, whether they're biotech, they're pharma, try to bring things to market, they want to try to do their trials as quickly as they can, get a result as quickly as they can, and then get a signal that it's going to work and be able to sell that medicine. So with that in mind, what's tended to happen in our field is that most of the drugs that have been developed overall, even including teplizumab, have really been aimed at stage 3, so that what we used to call new onset diabetes. And that's sort of where we're starting as well. Those study subjects are relatively straightforward to get. The trials have been very well designed and understood, and the rest. And we're hopeful that by selecting for DQ8, selecting the patients we're going to treat, we can see some good results when we do those trials. So our aim with that phase 2 study I was mentioning a few moments ago is to start there. And that's where that study would be in people who've been diagnosed, and usually within about three months from diagnosis or so. Now, that being said, we fully recognize, as all of you do as well, that going earlier would certainly be better. And the hard part of those studies, having been one of Aaron and I both participated in the teplizumab studies that have gone on. They've been conducted at the Barbara Davis Center as well as other places throughout the US, et cetera, is that the TN10 study took 10 years. So we can't, from a company point of view, do something that's going to take that long. Now, some of those studies can be done quicker now for lots of things that are changing in the field. Just the ability to have more people screened and know that they have autoantibodies that they could fit. But from a business point of view, it's pretty much a non-starter to say that's your first indication you're going for, as much as we would all like to do that. We're going to try to look to leverage in every way we can to get those kinds of studies started sooner than later in terms of the company and the funding. But from a marketing point of view, we have to start at stage 3 for the most part. And so that's sort of really the idea. Teplizumab is just a very unique sort of situation because, as people who may know about the drug, it had been in 2 large phase 3 studies and actually, wasn't successful with those studies for a whole bunch of reasons that we can talk about if you want. But the TN10 study, which was a relatively small study taken up by TrialNet, actually not by the company, and by Kevin Harrell, the investigator, the principal investigator of that, we were able to show that if you treat stage 2, you can actually have this benefit that looks really good. So we're hopeful that as companies start entering into this and realizing that these things are important, we can get them interested in starting to do those and launching those earlier in the process. But it's going to be an education process on every level, not just with all the people in the field and our colleagues as well, who aren't necessarily ready to screen everyone for antibodies, but also with the with the other pharma and biotech and investor community to realize the importance. The argument that we make, which we think is a good one, is that although there are a lot of people who get diagnosed with diabetes in a given year, 50,000, 60,000 or so, there are probably 1.5 million who are on their way to getting diabetes. And that's a really big market. So that is something they hear. So we're working on it, but it's not something that you get going overnight. Does everyone know what stage 2 is, by the way? I sort of was assuming that, but let me just explain.
Will you dig into those stages just a little bit more, Dr. Gottlieb?
Sure. Okay, so this is some work that we've all developed over the last few years. And I think this is an important piece because it is something that the regulators, the FDA in the US and the EMA in Europe are actually now understanding. So the thought is that, look, one of the things that we've been fighting against, and even with the things that we've been talking about tonight, is that when you're diagnosed with diabetes, you've had an autoimmune process going on for a bunch of time. We measure these autoantibodies, and we know that in some individuals, they may have had autoimmunity going for 2, 5, 10, 20 years, perhaps, depending on the individual. And to wait until their blood sugars go up sort of is giving away a lot of the beta cells in your pancreas. So the thought was we should really try to define the disease when you get to a point where we know that things are really going to move in a direction that's not so good, that really will lead to those blood sugars going up. So the way these definitions have been made is that stage 1 is when you have 2 autoantibodies. Okay, so it's not just one, not one to just insulin or to zinc transporter 8 or GAD or IA2, the 4 that we traditionally test, but you have to have 2 of them, and it doesn't really matter which 2. And at that point, you're considered stage 1. And if you're under the age of 20, there's pretty good data to say that you're going to get diabetes. It's more a question of when. If you're above the age of 20, that's less clear cut, but under the age of 20, the data is pretty good. Unfortunately, but again, it gives us an opportunity to change the course of the disease, as we saw with teplizumab, and hopefully may see with the Batacept, the drug that we're doing in TrialNet, and we'll report results on next year. So relatively, that's sort of thanks, perfect, thanks, Aaron. So if you look here, both Aaron and I we're really fortunate to work with George Eisenbarth, who really pioneered this sort of definition of the pathway to diabetes. And so this bottom half of the diagram really sort of is a takeoff of that original formulation, really, if you will. And the idea is that we're born with a lot of our full mass of beta cells, 100%. The genetic and environmental risks, there are both of those there. It's not just the genes we've talked about tonight. There are other things that act upon them. But at some point, things turn on and the immune system changes and you start losing beta cell mass. And obviously what we've been talking about with the new onset is that symptomatic point as you move to the right. So the stage 1 that I just mentioned a moment ago is beta cell autoimmunity, 2 antibodies, but importantly is noted there that you have normal blood sugars. You don't have high blood sugars yet. So you're considered pre-symptomatic. Stage 2 is the same. You can have the 2, you could have a few more antibodies, but at least the 2. The change is that now you have some, when you do a glucose tolerance test, you see some blood sugars that fall between the cracks that are not diabetes, but are not normal. They're sort of in between. And that's what we call the dysglycemia or impaired glucose tolerance. And once that happens, that's the stage where teplizumab seems to have been beneficial. So again, we would like to initially do the stage 3, move to stage 2, and I think eventually move to stage 1. One of the advantages of our therapy is we believe it's going to be safe. It's an oral medicine, which makes it easy to take, which means we could start very early in life and have people take this and just never really get the disease process going, hopefully arrest it really at the very beginning if we can get there. So that's, I think, where we would direct our thoughts and all the rest with how the drug would work, and also where we sort of are in the field.
Thank you for digging into that a little bit more, Dr. Gottlieb. And kind of a follow-up question on that, would there be possible synergy with teplizumab? Because it seems like your therapy would work best, like you just said, ahead of that broader T-cell response that would be then targeted by teplizumab. And Dan, feel free to jump off and clarify if I didn't ask that correctly. Dan is our research information volunteer on our board, and started his career in type 1 diabetes research and is a PhD and a wonderful asset to the organization.
Aaron, you want to go?
Sure. So actually I'll start and say, everything I know about diabetes and autoimmunity, I think even at stage 3, we're going to do well. I see autoimmunity as a threshold, and by genetically selecting, homogenizing patients that have their disease going through DQ8 and blocking that, I remain very positive that we're going to do good. Teplizumab works a little differently. This is more like induction therapy in cancer. It pretty well targets a molecule, CD3, on all T cells, those involved in diabetes, those involved in vaccination and in cancer, and those get removed from the peripheral blood and circulation. And so I do think there can be some synergy. I would actually consider using teplizumab first, coming back in, so you can't re-educate and reactivate those, say, insulin-specific T cells with the medicine that we're developing.
Yeah, and I think one thing to add there is that with any of the drugs that we're talking about, including teplizumab, it doesn't work for everybody. And the thought is that maybe that what we do need is really these combinations of medicines. So it is something that people are actively thinking about. And I think, as Aaron was saying, teplizumab as an initial drug with our drug following would be a very logical combination for someone who has DQ8. So I think that's something that people are moving towards and are thinking about as next strategies to try to take what we have as a good effect and actually make it even better.
Hearing that, and of course, going back to the individualized approach of that, right, and seeing what works best for that individual. We're so lucky that we have such really visionaries joining us this evening. And so, Lisa and Aaron and Peter, I'd love for you to look into your crystal balls that you all have and see if you can share with us what you think type 1 diabetes will look like in 5 years, what you think that'll look like, and maybe even more specifically, what you think JDRF's role in that might be.
Aaron, you want to start or you want me?
You get to go first. I went first last time.
Okay, fair enough. All right, so let's see. So the worst thing I think that when my daughter was diagnosed way back in '92, I think the worst thing that we ever, she was 2. The worst thing we were ever told is, oh yeah, 5 to 10 years it'll be cured. Don't worry, don't worry. So,
Right.
my wife is next to me. She corrected me. They said 5.
Promise 5.
Okay.
Hi, Gabby.
So I always am leery when I saw this question that they wanted us to answer. So let's see. I think we are in an interesting time. The hardest part is that clinical trials take a while. And I think if we were doing stuff in the lab, we can do an experiment in a few months and stuff. But if we're trying to do things in clinical trials, they just do take time. So over the course of the 5 years, I think, for example, our drug, if everything worked right, the type 1 diabetes drug, the DQ8 drug, at that time frame, if we hit on all cylinders, and so far we're doing pretty well that way, and things work out the way we're expecting, we would have an approved drug. So that would, I think, be a really important, piece to the field, and certainly for our company, and again, for the other approaches we want to take, not only for celiac, but for other autoimmune diseases as well. So that's my hope. That's my, why Aaron and I are plugging away at this. In terms of where the field may be, teplizumab is before the FDA, and they'll decide in the summer whether or not they'll approve it as a therapy. If they do, which I think would be a nice thing, but I'll tell you why I'm a little cautious about that, it will change the field. If we have an approved drug, again, this isn't the easiest drug to give and all the rest, but if there's an approved drug, suddenly the rest of pharma and biotech gets interested in our area, and that will be very, very helpful. Both Aaron and I have done this for a while, and there are times that diabetes has been the flavor of the month, and there have definitely been times when we have not been. And they're probably more of the latter than the former. So it would be really nice to be sort of in the hot seat and having people coming to us about doing things and being interested in what we want to do. So I think that could be very, very helpful. Now, the reason I'm a little, I don't know if skeptical is the right word, but a little bit cautious is look, that, again, we did the trial, there was only 70 patients in it. Typically, the FDA awards these kinds of designations for really large trials. So again, they're using a lot of data from the other trials that showed safety and the rest, and actually did show efficacy depending on how you define it. So we'll see. We'll just really have to see where that sits. But I'm very hopeful that we'll find other medicines that are showing effect. We already know that in the new onset setting, there are 6 drugs or so, 7 or so. The New England Journal article a week or 2 ago, for those who saw it, showed that there was some benefit from a anti-TNF medicine, the type of medicine that's been used for rheumatoid arthritis. And we showed in the diabetes that it could be helpful too. So I think that there's more of those to come, and as we start to apply them earlier in the disease, as we just looked in that graph, we're going to hopefully get even better results than we do when you do it later in the disease course. So I think that's really going to be a helpful thing. I'll add one last thing before I let Aaron add, is that I think JDRF is really critical to all of this. So the discussions that are going on right now, the FDA in the US, for example, is really trying to look at how are they going to define what it takes to allow a drug to move forward in our space. What's the criteria that a trial needs to meet? And JDRF has taken a really lead role in helping to define that discussion, to bring a lot of us together with the FDA to talk about that, to get everyone on the same page, provide them with the best evidence, both from academia and industry, so that we can really know that, okay, this is what you need to do to get a drug approved, and you hit these targets, you're going to get the approval. Right now, it's a little up in the air. It's not 100% clear what they would accept. And that obviously makes it hard for companies like ours and even larger ones to move forward in the area. So I think that type of work and the work that JDRF has done with now working towards payers, which will be really important, so that if we do get these drugs approved, that people will actually pay for them. And lastly, the special diabetes program, the SDP, to continue the funding for that. A lot of the work that we've done, a lot of the things that have led to where we are today have really been related to that funding. And right now it's always in jeopardy. But certainly, everyone's support for that would be really important, and JDRF has been critical. So I'd sort of leave it at those pieces, and then I'll let Aaron take over, and then Lisa can really tell us where it'll go.
All right, so I too remain very cautiously optimistic. Within 5 years, we'll have an approved medication, personalized therapy for type 1 diabetes, IMT-002. So if you're newly diagnosed, you have DQ8, you can take a medicine, and not that we're going to take you off of insulin, but life will be a lot easier to live with diabetes. I also think, given everything that's gone on, the success and momentum we've had, we'll have a drug for celiac disease in that time frame as well. It should work, and it should work very similarly to type 1 diabetes. I really want to see diabetes cured. I can't sit here and tell you that's going to happen in 5 years or even 10 years. I was diagnosed in 1992, and I was told too, in 10 years you won't have to live with this. Well, I'm now living with it as a patient, a physician, and unfortunately as a parent too. I think the findings of teplizumab helped me sleep at night a lot better. Those are the most clinically meaningful results I've seen in my entire career. We can intervene in the disease and keep people from living with diabetes. I know the New England Journal article says 2 years. Follow-up data show at least 3 years, which is great. 3 years without this disease is phenomenal. I think the goal for the field is, how do we make that better? How do we take 3 years and make it 5 and make it 10? How do we get diabetes to know type 1 diabetes not to be juvenile onset, but to be adult onset diabetes? How can we delay this and get it out of childhood, where it is, it's a lot harder as a child than it is as an adult. So I think that is the goal for the field. The JDRF's role, the JDRF does a lot. It has adapted, obviously, research funding, the T1D fund to really catalyze innovative research. I think their next goal is, doing these prevention trials in stage 1 and stage 2. It's challenging. We have plenty of investors that want to give us millions of dollars to do studies in new onset disease and get a drug approved. They don't want to wait 4 or 5 years to see results. That costs too much. And I see the JDRF's role as filling that gap and helping fill that gap. And they've done this in the past, and I think they can do that in the ensuing 5 years to really help get those things done, because I think in 5-10 years, we're going to see diabetes delayed much more than 2 years. And once we can do that, it really, really does open up avenues for curing the disease.
And Lisa, we knew this was going to happen. We knew we were going to get to, the bottom of the hour and want 4 more hours together. We all called it. We should have put money on it. But will you share your perspective and then close us out this evening?
Absolutely. And both Dr. Gottlieb and Dr. Michels did such a great job of explaining JDRF's role and strategy. I'm not going to repeat all the great points that they made. I do think that we are more excited than ever, but definitely we learned from the mistakes of the past in terms of overpromising. I think we've learned so much more about how complicated this disease is and how hard it is to cure an autoimmune disease, that there's been a lot of humbling in terms of what we really can expect in terms of a time frame as short as 5 years. But I can say that JDRF is as committed as ever to working each and every step of that pipeline, including increased population screening to identify people who are likely to develop the disease. And that can't happen overnight, but it is hugely important, with 85 to 90% of new diagnoses not having a family history like those of us presenting today. So we are counting on everybody's ongoing support, a combination of enthusiasm and excitement and a healthy amount of patience. But I really do feel that things are accelerating at an incredible rate, and ongoing involvement at every level, from donating and encouraging others to join us, to advocacy for the special diabetes program. And again, work all along that pipeline is what's going to get us to preventing and curing this disease for sure. So I just want to thank everybody. I want to thank our amazing presenters for your time tonight and for your giving of your lives to this incredible progress, and for your commitment. Obviously, you've got skin in the game, but we are all so deeply grateful that you are devoting your lives to helping the entire T1D community in such an inspiring way. I want to thank everybody on the phone for your incredibly generous support and your partnership, and encourage you to stay with us. And together, I am confident that we will put this organization out of business, not in 5 years, but we will not find ourselves in the position I am today, where our children are saying 100 years ago, my grandparents started this disease. We can do it, and I'm really confident that together we'll get there. So thank you all so much.
Thank
Anna,
you, everybody.
Thank you all. Have a great night.
Thank you, and enjoy that the dessert course for those who still have that to look forward to.
That last bottle of wine.
Thank you. Hannah, will you share, you'll share my
Thanks.
email address with everyone in case people have follow-up questions, of course, right?
Yes. Yep. We'll be sending follow-up with contact information.
Thank you all so
Definitely.
much.
Thanks.
Good night.
Good night.
everybody.
Thanks, everyone.
Good night. Thank you.